Thank you.
Thanks, Harris. Glad to see you all. And if you have any questions during the talk, just feel free to stop me. And I'm going to share my, hopefully, screen. Give me a sec. There we go. You would think after so much zooming that I could do this more efficiently. Okie dokie.
That's good.
Great. So I'm going to talk to you today about some research that my students and I have been doing about Asian bittersweet. And one of the introduction sites for Asian bittersweet in North America was the Biltmore estate. And I'll talk to you about some research that we've been doing to reveal some reasons why this plant might be such a successful invasive species. And I just want to acknowledge others who have worked with me on this project. So my students, Jenna Hamlin, Aaron Mazer, and Andrew Flynn. And then my grant collaborator, Matt Eastep. Got a grant to work on 8 species. I'm mostly just doing this one, but we did apply for and receive that grant together. So I want to talk to you first about the Celastraceae or staff tree family to which Asian bittersweet belongs. There are about 1,400 species worldwide in this family. That's the latest number. And the habit of plants in this family, they can grow as trees, shrubs, lianas, and herbs. Lianas are woody vines, and a lot of plants in this family include woody vines. And the Asian bittersweet that we'll talk about today is a liana, is a woody vine. So some characteristics of plants in the staff tree family include the ovary being up above the petals. The flowers are in a little disc. The fruit type is a capsule. And the seed has this aril around it, this little surrounding fleshy part that derives from the peduncle, the lower part of the flower. This is similar to what you see around nutmeg, if you're familiar with that one. So this family of plants is most diverse in the tropics and subtropics, but it's found worldwide. So it's much more common in the tropics and subtropics than it is here in the temperate region. And some North American species that you might be familiar with, besides the native congener, American bittersweet, there's also the American strawberry bush, Euonymus americanus, which is a beautiful plant that makes its fruits in late summer. So let's talk about American bittersweet first. So American bittersweet, or Celastrus scandens, is the native bittersweet in our region. And it's ranked S2 and G2. So the S rank is the state rank, the G rank is the global rank. So the G5 rank means it's secure. So worldwide, American bittersweet is doing pretty well. In North Carolina, the S2 rank means it's vulnerable to extirpation. So there are fewer than 30 viable populations of American bittersweet in North Carolina, and they're not doing very well. They're threatened by things like development and by the Asian bittersweet. American bittersweet is a deciduous liana, so it loses its leaves in the winter. It is one of those woody vines. And it's different from the Asian bittersweet in that its flowers and fruits are terminal, so they're at the ends of branches. It's also different from the Asian bittersweet in that it prefers limestone-rich soils. So it doesn't do very well on clay-heavy soils or saturated soils. It really likes those limestone soils. It makes fruit in the autumn, and it makes red fruit that is really delicious to birds, poisonous to you, but delicious to birds. And so the seeds inside those fruits are dispersed by birds. Humans use this plant for holiday wreaths. It's used throughout its distribution in North America to make these beautiful holiday wreaths, and the seeds from those wreaths can germinate in the compost. Not a problem if your wreath is from American bittersweet. Definitely a problem if your wreath is made with Asian bittersweet. So let's talk about Asian bittersweet, Celastrus orbiculatus. So this is an invasive exotic plant that is not native in its New Zealand and North American range. So this is native to China, Japan, and Korea. It is also a deciduous liana. It's different from American bittersweet in that the flowers are axillary. So they're in the branching points instead of at the ends of the branches. So the flowers and fruits are found in there. And this is much more common in our region. So if you see a bittersweet, it is much more likely to be Asian bittersweet than it is to be American bittersweet, unless you're up like in the Craggies. That's where you find the American, along the Blue Ridge Parkway, there's a lot of American bittersweet. It has really high seed production, higher than that of the American bittersweet, and it's often mistaken for American bittersweet. So you can see when the leaves have died back and you're just looking at the fruits, if you look at them side by side, the difference is pretty obvious. So the American bittersweet is much bigger fruit and fewer fruits, and the Asian bittersweet has more numerous fruits, and they're not just at the tip of those branches. Unlike American bittersweet, Asian bittersweet is not very particular about edaphic conditions. So it can go in a broad range of soils. Like American bittersweet, it can be spread by birds, and Asian bittersweet is also spread by humans who make wreaths and then compost those wreaths or put the wreaths out in the trash. So Asian bittersweet was a horticultural introduction. It was brought over from Asia in the late 1800s as a beautiful ornamental species with the intent to grow it in some gardens in North America. And there were 3 original sites of North American introduction, and those introductions seem to happen about the same time. So those 3 original sites were the Arnold Arboretum at Harvard University, and the Arnold Arboretum grows plants in their site, and they have a herbarium where they keep records of the plants, and they kept a good written record of this plant. And not only were they growing it there, but they were supplying it to nearby households, wealthy households in the region around Harvard University. The New York Botanical Gardens also brought over Asian bittersweet. They did not intend to put that out into the wild, but it escaped from their viticetum where they grow vines around 1898. They documented that escape and documented the spread of Asian bittersweet in the region around the gardens. And then this was also introduced as to the Biltmore estate. The Biltmore estate planted it around the grounds, and they also sold it from their nursery. So they did retail and wholesale distribution of this plant. When I first moved to Asheville 14 years ago, they were still selling this plant from their nursery. And they also sold it through catalogs. So they sold it throughout the United States. And the phenotypic similarities between Asian and American bittersweet meant that these escapes were not well documented, and naturalization was not really, land managers weren't really on top of the escape of the species into the wild because they didn't notice it for a bit. And here's a quote from the Southern Appalachian Highlands Conservancy. So Asian bittersweet can now be found in many eastern states, but the area around Asheville, North Carolina might win the prize for today's largest and most insidious bittersweet infestations due to successful marketing by area nurseries in the early years of its introduction. So the first part of this is uncontested. We have a much higher density of Asian bittersweet than other areas of North America. The second part, maybe this was from marketing. Maybe we're just a good place to grow. Okay. Here is a page from the 1907 Biltmore Nursery catalog showing that you could purchase the native bittersweet as well as Asian bittersweet. And the Biltmore estate originally got their bittersweet from Japan, that part of the region. So Asian bittersweet, you might know, is a pain in the neck to eradicate, and it does not great things to North American ecosystems, including shading. So these woody vines climb up trees and close off the canopy, which is really bad, especially for understory herbs, native herbs that are used to sort of a dappled canopy, have a hard time growing under Asian bittersweet. Asian bittersweet can reduce tree growth and girdle trees. So you can see here is a picture of some Asian bittersweet in Maine, and it's girdling the tree, it's squeezing it, it's reducing flow and movement up that trunk, and ultimately reducing tree growth. One of my colleagues, Jonathan Horton, and one of our students did a study here on the UNCA campus and showed some reduced tree growth also. Asian bittersweet is also not great because it makes a really strong connection from one tree to another. And if there's a windstorm or an ice storm and one tree falls, it can drag its neighbors along with it. One of the most interesting insidious effects of Asian bittersweet, though, is hybridization between the Asian bittersweet and the North American bittersweet. There was a study about this that came out in 2015 that used a few genetic markers to show that this was happening, but the morphological hybrids, so the intermediate looking phenotypes of these two plants, were first reported actually in 1947. And you can see how that might happen if you look at these two distribution maps. The top distribution map with the green is the distribution of American bittersweet, the native bittersweet, and the bottom distribution map in blue is the distribution map of the Asian bittersweet. And in many places, including along the Blue Ridge Parkway, these species co-occur and readily hybridize. They're flowering at the same time. They share pollinators. They can also do wind pollination. There's lots of opportunity for hybridization. So let's think about invasive species in general. And by invasive here, we mean species that aren't native and that are also causing some sort of ecological and or economic harm. So we know that there's ecological harm from Asian bittersweet. There's a lot of economic harm that happens to timber stands as well. So in general, invasive species have, there are 29 different hypotheses saying why invasive species become invasive, why they can get introduced to a new habitat, become established, and become problematic. The most common of these hypotheses are things like propagular pressure. So propagular pressure says that invasive species just make so many seeds that some percent of them are going to germinate and grow up and succeed. And so just by means of making a lot of offspring, that species can become established and problematic. Another hypothesis is that invasive species are really good competitors. And this has been shown with Asian bittersweet, that Asian bittersweet competes not only other species by shading them out, but outcompetes in side-by-side comparisons with American bittersweet. So it makes more seeds than American bittersweet. It can directly compete with American bittersweet for space and light. There's another hypothesis that's true of lots of non-native species like garlic mustard. Garlic mustard, an introduction from, gosh, I think Europe. Garlic mustard has a lot of natural enemies in its native range. Here in North America, there's nothing to eat it. So we call that the enemy release hypothesis. Folks thought that maybe Asian bittersweet was benefiting from enemy release. Maybe nothing ate it here, but actually deer really love to browse young Asian bittersweet. So that doesn't seem to apply. Disturbance tolerance. Asian bittersweet is really good at disturbances. It does quite well after fires or after tree falls. If there's a gap in the canopy, if there's suddenly a lot of light, Asian bittersweet can sit dormant in the soil. It can be in the seed bank for a decade or more and germinate when the canopy opens up, or it can germinate right away and stay tiny and then just really grow quickly once there's enough light. Things that have been less explored in Asian bittersweet include adaptation. So is Asian bittersweet phenotypically plastic? That is, can it respond differently to different light levels? Can it stay short if there's not a lot of light? Can it conserve its energy and get taller, wind around a tree when there's a light opportunity? Also, there hasn't been really an exploration of the genetic basis of adaptation in this species. So are some genetic strains better at growing than others? Has there been selection over time? So natural selection from the Biltmore estate introduction to prefer some genotypes over other genotypes. So that's what we've been focusing on in my genetics and field crew. And we've been focusing on two different hypotheses, two different research questions. So is Asian bittersweet phenotypically plastic? And does the plasticity vary by source? So are different populations around the Biltmore estate differently plastic? And second, what do population genetics tell us about the evolution of Asian bittersweet near the Biltmore estate? So, for instance, if you compare adult plants and seedlings, is the adult population more diverse than the seedling population? So there's some selection on who can germinate, some natural selection on that. Or is the adult population less diverse because birds are bringing in more seeds to the population? So not much has been done with the genetics of the Asian bittersweet. So that's what we've been working on. So I'll talk to you first about this phenotypic plasticity question. We used plants from Buncombe County in this experiment, and I'm going to talk to you about an set of experiments on plasticity that used plants from these three sites in Buncombe County. So the Biltmore estate is near here on this map, and we compared three different populations in here. So near the Biltmore estate and a little farther away for our plasticity experiments. Oops, there's a chat. Sorry. How would you? Oh,
That's a
yes.
question. We can address that later. I was
Yeah,
writing
no,
down.
it's cool. We can talk about it. So yeah, how, like, if there's a plant that is becoming invasive right now, like we know that there's this beautiful buttercup that's like showing up along creek beds. It flowers earlier in the spring. It's yellow and bright and beautiful. It's got variegated leaves and it's super nasty invasive. So it's like right now we're documenting new invasions mostly through citizen science. So sometimes a botanist will go out and notice something that doesn't seem to belong. And a lot of times it's things like iNaturalist. We've used iNaturalist to make those observations. That is really helpful to the scientific community to figure it out. The New York Botanical Garden escaped. The botanists from New York Botanical Garden and from the state of New York were walking around and noticed bittersweet that didn't look like the bittersweet that should be in New York State, because they have American bittersweet, but they just trained botanists notice. But I think that all of us, regardless of level of science training, formal science training, are capable of observing new things and checking to see if that iNaturalist is using, like I was working with a botanist from South Carolina last week, and she told me that when she's looking for these rare plants that she goes through iNaturalist and looks for people who've said that they've seen these rare plants, and then like actually goes to those sites and sees if they're really there. So put your observations in iNaturalist, help out the world.
iNaturals is definitely a very powerful tool. I'm glad to hear people are
Right.
actually using it.
Yes, and like if you want to go out and find the spring wildflower peak, look at last year's iNaturalist, and it will tell you when your species of interest was blooming and when you can get those really Instagrammable pictures.
It's all about the Instagram.
It is. It is.
Thanks, Jen.
So yeah. Okay, so my method, this is like a long text heavy slide, but we did two ways to look at phenotypic plasticity. We looked at adult plants and we looked at seedlings. So for the adult plants, we went out and found hundreds of plants from three sites and made these clonal replicates. So we cut them, so same genotype, same plant. These plants root really easily, unfortunately. So we grew them for a few weeks under common conditions and then moved them into these shade structures, representing an open forest canopy, a closed forest canopy, and a partially closed forest canopy. And these are just the light levels that we used there. And then we destructively harvested some of those clones at 0, 4, and 8 weeks to see how they might differ in how quickly they were growing and their leaf number and how big their leaves were, so sort of their photosynthetic potential. And then at the end of 8 weeks, we dried the plants to determine their mass. We also did this with seeds and seedlings. So we took fruits. Asian bittersweet does have to have a cold stratification period. So this is why in those invasion maps you saw it's not in Florida. It doesn't get cool enough in Florida to allow the seeds to germinate. They need a cool period before germination. So I put my plants in a cold chamber, for my seeds in a cold chamber for 2 months, and then we grew them in a growth chamber under controlled conditions and recorded when the seedlings first germinated, how tall they got, and the number of leaves weekly for 13 weeks. And at the end of that time, we determined the mass of the seedlings. So let me show you some of those data. So first takeaway point, bittersweet does have a lot of phenotypic plasticity, and the plasticity varies a lot by population. Okay, in this figure, on the y-axis is the leaf area, a measure of potential photosynthetic potential. And then here are the 3 light levels, so high light, medium light, low light. And here are the 3 populations. You can see that the one from near the Biltmore estate is bigger than the other 2, right? This blue line is consistently above the other 2. And this is the average leaf mass plus or minus 1 standard error. The statistics showed that these 3 colored lines, the 3 populations, were different, statistically different. So in fact, the plants from the Biltmore, from closest to the Biltmore, are bigger than the plants from these other 2 sites. And the plasticity also almost varied. So how much the plasticity is sort of an idea. So I was just comparing these parts of the slopes here. I was comparing the slopes of the lines, right? So how much the plants were able to adjust their size in response to the environment differed among those populations as well. And I'm just trying to explain here to you in green why I think that there's the biggest leaf area here in the partial canopy. So I think here the plants were pretty light limited. They were kind of suffering from light limitation. And I think this is a common thing that bittersweet does. It just sort of waits and stays small until the canopy opens. And here, sorry, that's here in the closed canopy, they're light limited. And here in the open canopy, I think they might have had too much light. The leaves looked a little bit yellow, and I measured some photosynthetic rates, which I'm not showing you here, but the photosynthetic rate in the highlight level was actually lower than the photosynthetic rate in the medium light level. Okay, so what about the final mass of the plant? So this is the whole plant mass. I find similar data for similar patterns statistically for if I just measure the leaves versus the below ground biomass. So I also here found differences among populations. Again, the plants from nearest the Biltmore did best. The light environments here were not statistically different. So the plant ended up making similar amounts of biomass in the different light levels. And that was just because they sort of switched their above ground and below ground allocation. So if there was a lot of light, if there was a medium amount of light, they made more leaves and smaller roots. And if there were high light or really low light, they made more below ground biomass, more roots. This Baldwin gap had the greatest biomass gain and plasticity, maybe making it more flexible. So this is the original Biltmore location. Certainly polluted with input from bird defecation of seeds from elsewhere, right? But this is probably the closest to the original introduction. Now the seedling responses did not vary by population. So if we started with seedlings, they depended on light level, but there wasn't any population level difference. So if we look at how many days it took them to germinate, I've mushed together all the populations in these data because they weren't variable. So here's the closed canopy, the intermediate light level, and the high light level. You can see that there is more germination in this open light and the darkness than there is in that intermediate light level. It's interesting because I just showed you in the previous graph that they were growing more as adults in that partial light, but they seem to germinate less. And this low light germination that I've been talking to you about promotes this thing that they call sit and wait strategy that a lot of plants do. So they germinate and then don't do further growth until conditions become better. If we look at the height of the seedlings, so how big they grow, as you might expect, they get a little bit taller when it's dark. I'm trying to reach up for that sunlight. Shorter when it's bright, and somewhere in the middle, statistically, in that intermediate light level. Again, the light environments had different amounts of seedling growth. The populations did not. I've collapsed all three populations into each bar because they did not differ at all. So this is kind of worrisome from a land management point of view, or from a try to clean up your own yard point of view, is that these young plants are growing pretty well under even low light conditions. So they are able to become established in intact forest ecosystems. So forests that aren't partially cleared or forests that aren't managed, these bittersweet vines can get pretty well established in. So let me tell you a little bit about the genetics now. So when I started to examine, I actually collected the samples for this genetic study 10 years ago. And I realized after I collected hundreds of samples and put them in my freezer that the genetic markers that existed for this species were rudimentary and did not amplify consistently. In 2015, they published 2 more genetic markers for this species. To do a good population genetic study, you need 7 or 8 genetic markers, so 7 or 8 spots in the genome to look at for variability. 2 doesn't do it. The folks who published the 2 genetic markers, that was useful in that those 2 genetic markers can also distinguish between the 2 bittersweet species, the American and the Asian bittersweet. So they use this to support their ideas about hybridization, but it doesn't tell you anything about variability within a population. And so I realized that I needed to develop more ways to measure population level diversity, and that has an upfront cost of about $4,000, which I did not have. So my collaborator, Matt Estep at Appalachian State, and I applied for a grant from the Great Smoky Mountains Conservation Association and got that grant. And the $3,000 to $4,000 that I mentioned gives you raw data that you can use to develop these population markers, and it gives you raw data for 8 species. So we applied to do this for ramps and sochan, which is another economically important species that's used by Eastern Baron of Cherokee, also for ceremonial and edible purposes. And we did bittersweet, and then we did 5 imperiled, highly imperiled species that are within the Great Smoky Mountains National Park. So my goal with this was to create markers that could show differences in, okay, bear with me, I'm going to give you a lot of words, but we'll define them. Intergenic regions of the genome, which are non-coding and highly variable. Okay, genome, all of your DNA. So if we think about all of your chromosomes, right? Chromosomes have DNA organized with proteins. So your genome, all of your DNA, bittersweet's genome, all of its DNA. If we look at the chromosomes of you or the chromosomes of bittersweet up close, we would find that there are these genes and then these spaces between the genes. Genes are coding, and by that I mean a gene directly affects a phenotype. A gene directly affects a trait. For instance, you have 2 copies of a gene that controls whether or not you can roll your tongue, right? So if you can roll your tongue, you have at least 1 copy of the dominant version of that gene. If you can't roll your tongue, you don't have any dominant copies. Earlobes, right? You have 2 copies of that gene, one from your maternal parent and one from your paternal parent, that control whether earlobes are attached or not, right? So those are genes. Genes affect phenotypes, affect traits. Intergenic regions don't. Intergenic regions might control genes, or they might be just non-coding meaning they don't show up in the phenotype, silent things. Now, even though these intergenic regions don't control phenotypes, don't control a visible trait, they're dragged along by evolution. And so they can tell you something about what's happened to that organism. They can tell you if that organism has undergone natural selection. They can tell you whether mating in a population is random or not. So is there some selection for who's mating with whom or not? These intergenic regions can reveal that information, even though they don't control visible traits or phenotypes or morphology. So the most variable parts of an organism's genome, of all of an organism's DNA, are these intergenic regions, because they don't control traits, right? They don't affect the appearance of an organism or the behavior of an organism. So they're sort of free to vary without hurting or helping the organism. So they tend to be more highly variable than other parts of the genome. And I was focusing on these regions called microsatellite loci. So these are parts of the genome where the nucleotides, the A's and T's and G's and C's that make up DNA, where the nucleotides repeat. And they repeat not because they're doing any function, but because the enzyme in your body and my body and body of Asian bittersweet makes an error when it's copying the DNA. So it sort of stutters, right? So it gets stuck. So here we're looking at a region of nucleotide repeats where there are 1, 2, 3, 4, 5, 6, 7 repeats of that CA motif, that CA pattern. And on the complementary strand, right, this is the C matches with G, A matches with T, 7 repeats of that. So all you have to know from this is we're looking for parts of the genome where there are repeats in intergenic regions that won't code for things, because these repeats can tell us differences, even about, like, you and your sibling have guaranteed multiple differences in your microsatellite loci. In like crime scene DNA, they use microsatellite loci because they're so variable. They can tell you really fine scale genetic differentiation. Okay. So what did I do in this screening for microsatellite loci? The expensive part was this first part, sequencing the nuclear genome of one Asian bittersweet plant to find its repeat sites. So I hope you're already skeptical, like you just did one plant. That doesn't tell you anything, right? Why did I only do one plant? Because one plant, well, 8 individuals of different species are $4,000, right? So I cannot do this for hundreds of plants. So this is the representative sample plant that I'm going to use to test, like, are these repeats found in other bittersweet plants? And they should vary in how many repeats. And I'll tell you more about that in a sec. Okay, so once I got this sequence, the sequence is literally like a readout, A, T, G, C, blah, 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 really long genome readout. And then I use a computer program that searches through all those letters and looks for the microsatellites, looks for repeat regions, areas where that enzyme that copies the DNA has stuttered and made these nucleotide repeats. And from that, I use another computer program that designs a complementary piece of DNA to go upstream and downstream of that region. So to sort of bookend that region of the genome so that I can make copies of it, enough copies of it to be visible. So I'm designing these primers, these short pieces of DNA to stick upstream and downstream either side of the microsatellite and make copies. And then I've got to check, like, do these really work on other plants? I know it worked on the sample plant. Would it work on other plants? Do they amplify consistently? And do they vary from plant to plant? If they don't vary, it doesn't do us any good, right? If we're trying to figure out differences within a population, no good. Okay, I'm just going to break this down for you a little bit more, and then I'll tell you the answer. So we can look at these microsatellite loci to figure out how long the nucleotide regions are. Here's my example I showed you before. So here is a microsatellite locus, just a microsatellite place in the genome, microsatellite place in the DNA of this bittersweet. It's got 7 repeats. So I'm going to make copies of this region using PCR or polymerase chain reaction. Use the primers that I designed with the computer program that looked through the genome of that one sample plant and found the repeat region and found the DNA that was beside the repeat region. So we're going to stick little DNA pieces, little primers, to either end of the microsatellite so that we can copy the in-between part. I add a fluorescent dye to one side of this, to one primer. One of the bookends has a fluorescent tag on it. So every time we copy that microsatellite will have a fluorescent tag on one side. Again, the number of microsatellite repeats will vary among taxa, so it would vary between Asian bittersweet and American bittersweet, and also among close relatives. So let's say this is the parent, 7 repeats. Its babies might have 9 repeats or might have 5 repeats, because when this parent makes an egg or a pollen grain, right, its enzyme is likely to make an error when it's copying the microsatellite region to go into that egg or into that sperm. The enzyme that copies the DNA doesn't do well on these repeated regions. It doesn't copy with fidelity on those regions. Okie dokie. So we use primers and the process of polymerase chain reaction to make enough copies to visualize it. And then once I've done all that in my lab for multiple plants, I send the fluorescently tagged microsatellites to North Carolina State University Genomics Laboratory, and I guess I should say, I mix it first with a size standard. So I mix the DNA with the size standard. I mail it to them, and they run it through this cool vertical gel with a laser at the bottom. And the laser scans for the color of the size standard and the color of the microsatellite. And then I put this picture into another computer program that tells me exactly how big that microsatellite region is. And from that, I can figure out exactly how many repeats are in that microsatellite region. And I can compare individuals, because individuals with more copy numbers in common are more closely related than individuals with fewer copy numbers in common. Okay, so here's why I'm so far on this DNA work. I have found this semester 10 novel, brand new to science, polymorphic, meaning they vary among individuals, because like I said, if they don't, we don't care. But novel polymorphic work more than 90% of the time with students doing the work, so I feel good about that. Microsatellite loci, this is like boring to you and exciting to me. So these are the repeats. So I found an AG repeat. You can see there are a lot of A's. Here's one that's just 2 nucleotides. Here's a tetranucleotide repeat, dinucleotide, trinucleotide, and this is the actual sequence of the primers, the DNA that's going to flank the microsatellite. This is the microsatellite, flanking, and this is just the temperature I have to set the machine to. So that was exciting. So then I decided to try to apply those microsatellite loci to some of my plant samples from 10 years ago. And when I was first collecting plant samples, I collected them in radii out from the Biltmore estate. And so I just started with the first radius, and I asked myself, is there a difference in diversity between the adults and the seedlings in this area right by the Biltmore estate? And so I tested them with the adults and seedlings. I started with 28 plants, so not very many plants. With those 28 plants, I did not find any difference in genetic diversity of adults and seedlings. So things that I had collected as big adults and things that I had collected as little sprouts. No genetic diversity. My caveat is that this is a really small sample size, so I don't know if this is a real pattern or if it's because my statistical power is really low with only 28 plants per age. So a couple things. So if this pattern is true, like maybe the natural selection that happened early on in the invasion process. So 120 years ago, not currently happening, maybe. I am curious though, because I think that even if the natural selection has happened, that we're on major migratory pathways here in Asheville, right? And I think that, and I have observed flocks of migratory birds eating Asian bittersweet fruits, right? So I think that even if there isn't any natural selection happening, there should be some sort of evolution through migration of these plants as seeds carried by migratory birds. But I haven't seen that yet, but stay tuned. Give me like a year or two and I'll come back. So what are we doing still? I'm working to develop additional microsatellite loci. Ideally, I would publish this with 20, not 10. People have published with 10, but I'd like to get to 20. I also want to check them against some pure American bittersweet to see if they cross amplify. So sometimes within one genus, within Celastrus, you can get both to amplify. That's what that Zea paper did when they were trying to check for hybridization. I want to complete my seedling and adult analysis of the genetics and then go back to my giant 1,000 plant freezer set of plants and see if there's any spatial difference in genetic diversity. We saw a difference in performance and plasticity, but if there's any genetic difference. So I do have 100 sites total, and I do have 10 plants per site, so that's 1,000. Okay, I want to end by thanking my faculty collaborator, Dr. Matt Estep at Appalachian State University, my student researchers, Jenna Hamlin up here, Aaron Mazer here, and Andrew Flynn, who did a lot of the groundwork on this, and my genetics crew from my classes last year and this year, which did some of the screening with me to see, like, could we really get high success rates on this? Funding from the Great Smoky Mountains Conservation Association and the biology department here, and then logistical support for making those beautiful graphs from the North Carolina State University Genomics Laboratory. Thanks, and I'm happy to answer your questions. Stop sharing.
Great. Thank you so much, Jen. That was super interesting.
Oh yeah, you know what? I have not done hybridization tests, but if these new markers cross amplify with the American bittersweet, I think that I could do some hybridization work. I've been doing hybridization work on pitcher plants, and I have enough markers, enough genetic spots with that to figure out, like, are they early hybrids or later hybrids? So Claudia Prieto-Alciad, who talked last time at Science Tavern, is running that work with me. So I think that would be really great, because right now they can just say, oh yeah, it's got one marker or both markers, or that there's good understanding of hybrid or not, and there's not good understanding of where those plants are in their hybridization process. So early hybrids or later hybrids, or like a hybrid breeds back with the American bittersweet, or a hybrid can only breed with Asian bittersweet. It makes a big difference as far as management goes. And managers in North Carolina are really interested in preserving the American bittersweet because it is relatively uncommon in our state. Yeah, that's a great question.
Yeah, that's an important distinction to make, is that historical background to it, which is definitely challenging to establish from a baseline.
Yeah, I'd be interested too if like, so in theory, there could be hybridization even in spots without the Asian bittersweet, because the pollen can be windborne. So
Right.
that would be interesting to see like a degree of hybridization, because that paper also, I should say, only checked for hybridization along the Blue Ridge Parkway near Asheville, where you would expect it more, because again, we have a much higher density of Asian bittersweet than other places in North America. So maybe if you checked a lower density region, like down in Georgia, they have Asian bittersweet, but not as much.
Cool.
How was that genetics? Too much? Too little?
I loved it.
I mean, Harris, you better put your thumb up.
I feel like I was back in Elisa's class. I was like, I could tell she's taught a genetics class before. This is great.
We appreciate you explaining it.
Yeah, thanks.
Very much.
I'm trying to make genetics accessible so much. Yeah, I really admire journalists like the New York Times. I think does a really good job of explaining genetics when they talk about a genetic study. But it's I mean, I teach this and do this, and I have to read all the time to keep up with what's happening because it's really rapidly shifting.
I had a question just with genetics understanding, statistically speaking. So what some of your statistical claims were really close to that 95% confidence gradient. What do you do when you have statistical claims that are, something like 94.5% supported? Like, how do you kind of support those claims? Because it's like that 0.5% doesn't really matter, especially when you're talking about sample sizes, in the sub 100 range or sub 1,000 range even.
Yeah, it's a good question. For the plasticity studies, I did a power analysis on like a little preliminary study that showed that I had enough replicates. So I can't, sometimes I can say like, oh, I just needed more replicates, but I think that I am explaining it as nearly significant. And I think ecologists sometimes let you get away with like small, like 0.056 might be okay for ecologists. So, I mean, I'm sure there are confounding variables, like there was some insect herbivory a little bit, right? Like I started out with, I only needed really 6. I needed only 6 replicates because I did an initial measurement and then a second measurement that I destructively sampled and then a third measurement that I destructively sampled. So, and I had the different light treatments. But I did like 3 extras in case some died, which some did, and I just tossed the extra ones. I don't know, stuff happens in ecology.
Yeah.
Stuff happens in the genetics lab too, and I just like rerun the sample, but if it's a living plant, yeah, it's tricky, but I do think that ecologists have a lower standard.
Yeah, I mean, ecology is a general field, you're kind of looking at the greater picture rather than a specific niche or something like that.
For sure.
It's interesting. Well, this was honestly a great, I've wondered for a while now about that process of identifying microsatellites. So this was greatly informative to me. So I appreciate this talk immensely. I have another specific genetic question, but maybe I'll hold off maybe when we catch up sometime soon. But I want to thank you for giving us this talk. I really appreciate it. And good to hear about your other work that you've been working on, and love to see what you do in the future. So don't be a stranger.
Thanks. John, thanks for the word understandable. I appreciate that.
Yeah,
struggle
that
with
was
this one.
excellent. The breakdown was much appreciated.
Definitely.
Yes,
Awesome.
thank you.
All right. Thanks, friends. I appreciate you all. And I look forward to seeing Science Tavern back in person outside.
Yes, we
Or
do
maybe
as
just
well.
this virus could leave and then we can go inside again one day, but maybe not. Yeah. Doesn't seem to be happening anytime soon.
Thanks for
It's
all your
okay.
work.
Yeah, thanks. All right. Thanks for inviting me, folks. And I hope you all have a good evening.
Thanks, Jim.
Take care.